All right, welcome everybody. This is literally the fifth time I'm recording this, so hopefully fifth time is the charm. It's also the third time I'm making this joke, so. Well, welcome to the tricuspid valve actively maladapts, a perspective from the proteomic to the tissue scale. I'd like to thank my students as well as my co-authors who have been incredibly helpful with this project. The problem I'm trying to address and I'm trying to understand is that of tricuspid valve regurgitation. Tricuspid valve regurgitation describes leakage of the tricuspid valve between the right atrium and the right ventricle. About 1.6 million Americans suffer from tricuspid valve regurgitation, and currently the treatment options are limited as well as suboptimal, with 10 to 30% of treated patients experiencing recurrence regurgitation within a couple of years of surgery. In 85% of patients, tricuspid regurgitation is functional or secondary, and that means that the valve itself is considered intact, and the reasons for regurgitation are considered external. In most cases, that means that left heart disease has caused right heart disease, during which the right ventricle dilates, pulling on the valve leaflets, and therefore causing regurgitation. Accordingly, the valve is treated, meaning that in tricuspid regurgitation, surgical treatment is primarily annuloplasty, in which the valve is cinched, but the leaflets themselves are not treated in any way because they're presumed intact. The objective of this work and sort of the larger project that I'm working on is to move away from what I call the passive flap approach or paradigm, in which the valve itself is treated like a check valve, like a mechanical valve whose tissues are not alive. But instead, I and others believe that the tissues are alive, they're active, and therefore we should treat them accordingly, and our treatment options should also treat them like they're active tissues. And so here we set out to determine whether the tricuspid valve tissues actively respond to disease, or in other words, maladapt. To this end, we used an experimental sheep model in which we induce biventricular heart failure by overpacing the animals with about 200 to 220 beats per minute of excitation. Within 2 to 3 weeks, these animals develop signs of biventricular heart failure, and that includes ventricular hyperkinetics, reduced ejection fraction, as well as mitral and tricuspid regurgitation. Once the animals have reached their quantifiable endpoints, such as ejection fractions smaller than 30%, as well as significant tricuspid and mitral regurgitation, we sacrifice the animals, we explant the tricuspid valves, and in this case, we measure the area, width, and height of the tricuspid anterior leaflet. In these measurements, we find that actually within 2 weeks, the control animals in blue and the disease animals in red are significantly different. And specifically, we find that the area has increased by about 30% in just 2 weeks to 3 weeks of tachycardia-induced cardiomyopathy, or TIC, which is our biventricular heart failure model. That area increase primarily resulted from an increase in width and not so much height. And because reviewers asked, we also looked at whether this area was correlated to animal weight, and we're happy to report that it was not correlated to animal weight. We also measured the thickness by embedding these in paraffin and optically measuring the thickness from the annulus all the way to the free edge. And again, in red, we have the disease, and blue, the control animals. And we see that the thickness increases sort of qualitatively. Quantitatively, we see also an increase. We see statistically significant differences only at the free edge, and marginal differences, if I may use the word, in the belly. But if we group them together, we see overall a thickness increase of about 40%. So these leaflets increase in size and they increase in area. We also conducted planar biaxial tests. And here again in red, we see the disease animal, and blue, the control animal. We see these leaflets shift to the left, or their mechanical response shifts to the left. On the left-hand side, we see the circumferential response. On the right-hand side, the radial response. And we see two differences. In the calf stiffness, which is the stiffness after strain stiffening and this J-shaped curve has begun or has been initiated, we see a 30% increase in the stiffness in radial direction. In the toe stiffness, that is the stiffness down here, we see actually an increase in circumferential stiffness. And so we don't have data backing this up right now, but generally people associate this toe stiffness more with elastin, while this calf stiffness is more associated with collagen. But either way, we see a difference in the mechanics of these leaflets. We also looked at collagen content via hydroxyproline assay, and we find here that by the annulus as well as by the free edge, we see an increase in stiffness and nearly in the belly. And if we group them together, we see a significant increase of actually around 40% collagen content. And this is normalized by wet weight. We also conducted 2 photon imaging of these leaflets. We optically cleared them so we could look throughout the entire thickness. Here we have the atrialis surface at 0, the ventricularis surface at 100. So this is a look through the thickness. And for each thickness, we used image J to quantify the distribution of fibers. And these are the probability density functions basically shown throughout the thickness. So the mean direction is around 90 degrees, which is something you would expect. That's our circumferential direction. And we have dispersion around this direction throughout the thickness. We see a marked difference when we looked at these disease animals in that the mean direction hasn't changed, but the dispersion is actually much wider. And when we fit von Mises probability density functions to these histograms, then we see actually that the concentration parameter goes down, meaning the dispersion increases. It's a little counterintuitive near the annulus. So in addition to collagen overall being upregulated, we also see sort of a loss in microstructural organization. We conducted immunohistochemical assays in which we find that alpha smooth muscle actin is increased throughout most of the leaflet. And the way that these graphs are supposed to be read is in this top layer, we have the atrialis, bottom layer, ventricularis. So we have sort of a look through the thickness. Then we also differentiated between annulus, belly, and free edge. So this is kind of a transformed map of the leaflet cross-sectional area. And red means that there's more in the tick animals, meaning the diseased animals, than the control animals, and blue would mean more in the control animals. So we see overall, we have mostly red, meaning upregulation or increased activity of alpha smooth muscle actin, implying that valvular interstitial cells likely became activated and express this sort of myofibroblast-like character. We also see Ki67 being upregulated, a marker for cell proliferation, as well as MMP13, a marker for increased matrix turnover. So overall, the tissue we have shown increases in size, becomes thicker, becomes stiffer, collagen content increases, collagen becomes disorganized, and we also kind of see on the cellular level that there is increased activity. We also performed proteomic studies in which we identified several clusters associated with proliferation of cells. I'm not going to go into these details that are obviously multifold, but we uploaded the data to the bioarchive, and I would love it if you check that out. Overall, the model that we're adopting to understand what's going on is one that was developed by Del Bianco, and Levine is the co-author of this work, for the mitral valve. And here, the theory is that the combination of endo-MT as well as activation of valvular interstitial cells results in an overall proliferation of the tissue and a fibrotic response that then results in area increase, increased collagen, thickness increase, disorganization, as well as stiffening. So where are we going to go with this work? There's so much to be understood, right? Our study was not very mechanistic in the sense that we're just describing all the things that are happening to the leaflets that seem to imply the leaflets are active, they maladapt to disease, but it's a pretty complicated disease, a pretty difficult disease model. So our next focus will be to understand the stimuli. Are there pathomechanical stimuli? Are these biological stimuli? Are they both coming together? We don't know currently, but that's something we want to understand. We also don't know the consequences. Obviously, we can't all envision that a thicker, stiffer leaflet is probably not good for coaptation, but we don't know that yet for the tricuspid valve. And then overall, this is a really exciting topic because the idea that there is a biological issue with the leaflet itself and not just sort of an extrinsic secondary issue also implies that there may be new approaches to fixing the tissue, including pharmacological approaches that could regulate this maladaptive response if it was partly responsible for regurgitation. I would absolutely love it if you looked at a review article that I recently wrote on this topic, as well as this paper and this work that's been uploaded to the bioarchive. And with that, I think I'm basically at time. I'd really like to thank my students again, my co-authors, you for being here, BMES for organizing this and putting this together, and my funding sources for having enabled me and my students to conduct this research. I hope you're all safe, and I really hope to see you all soon in person again.